Greetings everyone, I'm Phehello Chauke and today I'll be presenting my MSc research titled Menopause-related changes in the gut microbiome and their association with cardiometabolic diseases in women from 4 sub-Saharan African countries, and I was supervised by Prof. Scott Hazelhurst, Dr. Louisa Olubayo at the back, and Dr. Dylan Magini, who's not here. So to get into the background, menopause is a natural biological transition that is marked by the cessation of menstruation for 12 or more consecutive months. It is characterized by the loss of ovarian function, which is then accompanied by a decline in ovarian sex hormones such as estrogen, progesterone, and sex hormone binding globulin. This transition has been associated with an increased risk of cardiometabolic diseases such as your hypertension, type 2 diabetes, hypercholesterolemia, and. So it has been hypothesized that the gut microbiome may be a mediator between menopause and the increased risk of cardiometabolic diseases. So the gut microbiome is a collection of trillions of microbes that colonize the gastrointestinal tract. It influences various physiological processes such as your immune function, your metabolism, and it is continuously shaped by factors such as your age, your hormones, your lifestyle, environment, etc. So research suggests that there's a bidirectional relationship between estrogen and the gut microbiome. So the gut microbiome contains what has been termed as the estrobolome, which is a collection of microbial genes that are able to deconjugate estrogen, allowing for its reabsorption into the enterohepatic and or systemic circulation. Estrogen, on the other hand, is able to maintain the gut balance by influencing intestinal barrier function and microbial composition. The aim of the study was to investigate the compositional differences of the gut microbiome between pre and post menopause and their association with cardiometabolic diseases in African women. So to get into the methods, this microbiome study is a sub-study of AWIGEN, which is a longitudinal study that recruited participants in wave 1 and followed them up in wave 2. The participants were recruited from 6 sites in 4 countries, 3 sites in South Africa, and then we had a site in Kenya, another in Ghana, and Burkina Faso. So this study semi-randomly selected 1,801 women from the AWIGEN cohort, and it leveraged AWIGEN's existing data on demographics, health data, and reproductive health data or health data, and the reproductive data. And then a microbiome-specific questionnaire was added to capture the lifestyle and environmental factors that may affect the gut microbiome. Following that, stool samples were collected for the microbiome analysis, and that complemented the blood, urine, and genetic data that was collected for the broader AWIGEN Wave 2 study. So the original data generation included DNA extraction from fecal samples that was then followed by shotgun metagenomic sequencing, and then an in-house Nextflow pipeline was used for the microbial analysis. Then taxonomic profiling was performed. So the menopausal status was self-reported, and the women were grouped as premenopause. So you have your regular menstruation, perimenopause, you've had fluctuating menstruation in the last 12 months, and then postmenopause, where you have not had any menstruation in 12 or more consecutive months. And then the criteria that was used for classifying cardiometabolic diseases is shown on the slide. And we chose obesity, hypertension, diabetes, and hypercholesterolemia. For the latter 3, medication use or a prior diagnosis by a healthcare professional also met the criteria. So to classify the women as with and without cardiometabolic disease, we used a composite score of 0 to 4. So a score of 0 was classified as without cardiometabolic disease, and a score of 1 to 4 was classified as with cardiometabolic disease. And so alpha diversity, which is within individual diversity, was measured using the inverse Simpson index, which accounts for evenness and richness, richness being the number of species present and evenness, the relative abundance of the species present. Then the women were stratified according to they were stratified by CMD status, and then the impact of the menopause and CMD was tested. Age and CMD were also tested separately as confounders. Beta diversity was measured using Bray-Curtis distance, and PEMANOVA was used to test menopause, cardiometabolic disease, and their interaction across univariate, additive, and interaction models. And then for differential abundant taxa between pre and postmenopausal women, a linear discriminant analysis effect size was used. So all statistical analysis were carried out on R, and multiple testing was corrected using the Benjamin-Hodgberg procedure. So to get into the results, from the 1,801 women, we excluded perimenopausal women just so we can see the distinct differences between women that have undergone menopause and those that haven't. And we excluded women that had undergone hysterectomy, since the cessation of menstruation does not necessarily mean the loss of ovarian function, because the main thing about menopause is the loss of ovarian function. And then we excluded birth control users to avoid confounding from altered sex hormone, therefore leaving us with 1,232 women, so 880 of which were postmenopausal. And so just to look at the participant characteristics, premenopausal women were younger than the postmenopausal women. And in the premenopausal women, about 39% had cardiometabolic diseases, and in the post, 74% were with cardiometabolic diseases. And looking just at the socioeconomic status, both groups were in the lower or the poorer class of socioeconomic class. And then to look at alpha diversity, overall, premenopausal women had a slightly higher microbial diversity than postmenopausal women. However, this was not statistically significant. And looking at the site-specific analysis showed like an opposite trend. In most sites, postmenopausal women exhibited higher diversity than the premenopausal women. Nairobi was the only site that mirrored the global overall picture. And then the site-specific analysis was only statistically significant in Agincourt. And then after stratifying by the menopausal status and by CMD status, overall women with cardiometabolic diseases had lower microbial diversity than those without cardiometabolic disease. However, within each group, there were no significant differences between the pre and the postmenopausal women. And then when we looked at CMD as a confounder, it revealed a statistically significant relationship between individuals with cardiometabolic disease and microbial diversity. Further, there was no interaction, between cardiometabolic disease and menopause. And when we looked at age as a confounder, there was also no statistical significance. And for beta diversity, the findings also reveal that the effects of menopause are not consistent across sites. So looking at two sites, Nanoro and Agincourt, Yes, looking at two sites, Nanoro and Agincourt, we can see that in Nanoro, there was a statistically significant menopause significantly affected beta diversity in Nanoro and showed borderline significance in Agincourt. And when we looked at Nanoro effects of CMD, there was a CMD also statistically significantly influenced beta diversity, and there was an interaction between menopause and cardiometabolic disease. In contrast, at the other sites, there were no significant effects of menopause, cardiometabolic disease, or their interaction. Just looking at differentially abundant taxa using, again, Agincourt and Nanoro, the distinct taxonomic differences varied substantially from site, because these sites, some of them are agricultural, some of them are have transitioned, are urbanized, and some of them are in the transitioning phase. So there were distinct taxonomic differences. So postmenopausal women were enriched with short-chain fatty acid-producing bacteria, which have been shown to play a role in metabolic health, while postmenopausal women were enriched with a mixture of beneficial and potentially harmful bacteria. And so just to get into the discussion, our study showed that cardiometabolic disease status emerges as a stronger determinant of microbial diversity than menopausal status. Geographic location also significantly influenced microbial diversity and compositional patterns, with substantial variation across the study sites. Taxonomic analysis revealed differential abundant taxa between pre- and postmenopausal women. So while menopause itself may not directly drive the overall microbiome composition and diversity changes, the taxonomic shifts between the pre and the postmenopausal women suggest that hormonal factors may still play a role in shaping specific aspects of the gut microbiome. Thereby, this suggests there may be a complex relationship between menopause, cardiometabolic diseases, and the gut microbiome. So to get into some limitations, the cross-sectional design of the study prevents establishing any causality between microbiome changes, menopause, and cardiometabolic disease development. Again, we could have included endocrine sex hormone measurements, because this would have better elucidated the relationship between hormones and the microbiome. And again, the use of the composite CMD score may have obscured some of the relationships between specific cardiometabolic diseases and microbial patterns, because now we don't know if what we're seeing, is it obesity, is it hypertension, is it diabetes? We just use the composite score. So just to conclude, to our knowledge, this is the first comprehensive analysis of menopause-related changes in the gut microbiome in African women. So our findings highlight the complex interplay between menopausal status, geography factors, and metabolic health. And it necessitates population-specific approaches when we study women's health, and it further contributes to understanding of women's health during aging in African populations. And I'd just like to thank my supervisors, all the funders of the study, community advisory groups, and anyone else that is involved in the study. It's a very big study.
Thanks very much. Are there any questions?
I think that this can be a good candidate for 10, 20 years, because there is well documented. There's nurses following you say when they follow the nurse 20 years before the cancer, after the cancer. So that can be a good candidate.
Okay, we've got one question. Yes, sir.
Thank you for the great talk. So I'm just wondering why you have used just one index for the alpha beta diversity. You could have used that indices. That would give you much more context to that.
Okay, so thank you for that. So yes, we did look at richness, evenness as separate indices, and then we also looked at the Simpson index and the I'm having a mind block, but yes, we did look at all the indices, and then I just chose inverse Simpson because I felt that since it accounts for richness and evenness, then we get the overall picture.
Any more questions? Are we good?
Thank you for the talk. You mentioned the overall study sampled at 2 different time points. Do you have any samples that have where the individuals are matched at those time points? Because that's been quite a big time difference between, so it would be interesting if you compare different sample points, especially if those women have developed potentially balloon. Like in that time had undergone menopause.
Okay, I think you're asking if because the parent study had wave 1 and wave 2, so the microbiome study is a sub-study of the wave 2. So when these women were followed up in wave 2, then that's when they collected the data. That's the data that we use for our microbiome study.
Okay, thank you very much.